everyone, welcome back to Sowkya Vision. Today we are going to see the erythropoiesis. Yes, in the last class we have completed the RBC, the site, production and morphology of RBC and fate of RBC importantly, and today we are going to see the process of development of RBC. Yes, so what is erythropoiesis? Yes, poiesis meaning the process of maturation. Yes, erythro means RBC obviously. Yes, so the process of from origin, development and maturation of erythrocyte, that is RBC, is known as erythropoiesis. Yes, the origin, development and the maturation of erythrocyte is known as erythropoiesis. Yes, now erythropoiesis, these RBC, the survival period of RBC is what? It is about 120 days, right? So the survival days, the lifespan of RBC is about 120 days. So after attaining 120 days, they get destructed in the spleen. Yes, the spleen is a graveyard of RBC, as we know. So after attaining 120 days, they get destructed in the spleen. So the new red blood cells are synthesized. Yes, on and of new red blood cells are synthesized. Attaining 120 days, they get senile and destructed in the RBC. Yes, so now let's see what are the sites and what are the at what stages, at what places the RBC are synthesized in during lifespan. So in the fetal life, in the embryonic stage, in the intrauterine life, there are three main stages in the process of synthesis of RBC. So what are the three main stages? They are mesoblastic stage, hepatic stage, myeloid stage. So what is the mesoblastic stage? So it is a stage at 2 months of intrauterine life. Yes, it is a stage of production of RBC, that is erythrocyte, at 2 months of intrauterine life. So from where these RBCs are produced in mesoblastic stage. So they are produced from mesenchyme of yolk sac. So in the fetal life, the mesoblastic stage, that is at 2 months of intrauterine life, the RBC are produced from mesenchyme of yolk sac. So the next stage, it is hepatic stage. Yes, in the hepatic stage, that is at 3 months of intrauterine life, the RBC are produced. That is, hepatic means what? Liver, spleen. Yes, the RBCs are produced from liver and spleen. In the myeloid stage, it is about 3 months, last 3 months. Yes, it is from last 3 months of intrauterine life. The red blood cells are produced only from red bone marrow. Yes, there are 3 stages in the fetal life, that are mesoblastic stage, hepatic stage and myeloid stage. Yes, according to the stages, they are synthesized in mesenchyme of yolk sac in the mesoblastic stage, in the hepatic stage, liver and spleen, myeloid stage, that is only from red bone marrow. Yes, in the newborn, the RBCs are produced only from red bone marrow. Yes, in the adult up to 20 years, yes, up to 20 years, the RBCs are synthesized from red bone marrow of long and the flat bones. What are the examples of long bones? The femur, the tibia and the humerus, radius, these are the long bones. Now what are the examples of flat bones? Like scapula, it is a flat bone. So the in the adult, up to attaining 20 years, from the birth to 20 years, the RBCs are synthesized from red bone marrow of long bones and the flat bones. After 20 years, yes, after 20 years, these RBCs are produced only from membranous bones. Yes, only from membranous bones. What are the membranous bones? These are vertebra, sternum, ribs, scapula and etc. So these are the sites and in the lifespan where RBCs are produced in different stages. So let's see the erythropoiesis. Now here we go. There are 6 stages. There are 6 main stages of erythropoiesis. Yes, what are these stages? They are pro-erythroblastic stage, early normoblast stage, intermediate normoblastic stage, late normoblast, reticulocyte and finally mature RBC. So pro-erythroblast. Blast means the process of development. Yes, the blast means process of developing stage. So proerythroblast. Yes, proerythroblast is the first largest cell in order to develop into different stages of erythropoiesis. So this is the largest cell over compared to all other stages. So hence it is known as megaloblastic stage. As it is a proerythroblast, it is the largest cell. Yes, largest cell, largest nucleus. So it is known as megaloblast. Yes, and next stage is early normoblast. Early normoblast. So the cytoplasm in the in early normoblast stains basophilic. So it is known as basophilic erythroblast. Yes, next the intermediate normoblast. In the intermediate normoblast, the hemoglobin starts appearing. See, in the first step, second step, there will be no hemoglobin. In the third step, there will be appearance of hemoglobin. So hemoglobin adds the acidophilic, yes, which gives the acidity to the acidic nature to the RBC. It is not matured RBC, but yeah, hemoglobin gives acidic character to the cytoplasm of the cell. So hence it is known as polychromatic or polychromophilic erythrocyte. Now reticulocyte. It is a immature erythrocyte. It is a immature erythrocyte and it is developed into RBC after removing complete reticulocytes. Yes. Now what are the stages? Once again, there are 6 stages: proerythroblast, early normoblast, intermediate normoblast, late normoblast, reticulocyte, and finally reaching mature RBC. Yes. Now what are the changes? Yes, in order to develop, there should be some changes, right? From proerythroblast to mature RBC, there are some changes taking place in the cytoplasm, nucleus, and overall the structure of the cell. So what are these changes? These are the changes. Reduced size of cell. Yes, reduced size of the cell. As I said, the proerythroblast is the largest cell compared to all other stages in the erythropoiesis. Yes, the from proerythroblast to mature RBC, the size of the cell decreases. Yes, the proerythroblast is about 25 micrometers in diameter. Yes, the diameter of the proerythroblast is 25 micrometers, whereas the diameter of the mature RBC, it is about 7.2 micrometers. Yes, it is about 7.2 micrometers. Now next is nucleoli and the nucleus. Yes, the peculiar feature of RBC is absence of nucleus, right? So the proerythroblast, from the proerythroblast to the mature RBC, there will be disappearance of organelles like nucleolus, mitochondria, and Golgi apparatus. Yes, and now there will be appearance of hemoglobin. As I said, the hemoglobin appear in the later stages of the erythropoiesis. It is absent in the early stages. Yes, it is absent in the proerythroblast and the early normoblast. It starts appearing in the intermediate normoblastic stage. Yes, and the fourth one is change in staining property. Yes, the cytoplasm of the cell. Yes, the cytoplasm of the cell priorly it stains basophilic. Yes, after formation of hemoglobin, which adds acidophilic, it changes its staining property. Yes, the staining property of the cytoplasm changes from basophilic to acidophilic. Yes, let's see the process of erythropoiesis. Yes, as I said, there are 6 stages in the development of erythropoiesis. So the first stage is proerythroblast. As I said, it is the largest in size. Yes, it is the largest in size. Hence, it is known as what? Megaloblast. It is the largest. See, this is the proerythroblast, early normoblast, intermediate normoblast, late normoblast, and the reticulocyte. Finally, reaching what? Mature RBC. So the megaloblast, it is the largest compared to all other stages. So it is the largest cell. As I said, the diameter ranges from 25 to 20 micrometers. Yes, the diameter from 20 to 25 micrometer. Now the nucleus of the proerythroblast, it is very large compared to all other stages. As the stages as the RBC, the cell get matured, the nucleus decreases in size. So the nucleus is largest in proerythroblastic stage. So the cell, the cytoplasm and nucleus and all other components of the cell are very prominent, yes, very abundant in the first stage. So the nucleus is very large, containing two or more nucleoli. Yes, this containing two or more nucleoli, very big cell. Yes, and the very big nucleus containing two or more nucleoli and the reticular network. So the cytoplasm is staining what? Basophilic. Yes, the cytoplasm staining basophilic. So here hemoglobin is completely absent. There is no matter of hemoglobin. It is completely absent, but it starts synthesizing in this stage. Yes, hemoglobin is absent obviously, but it starts synthesizing in this stage only. In the proerythroblast, hemoglobin starts synthesize, but it is not appeared in this stage. Yes, now the next stage is what? Early normoblast. After this, the early normoblast is started. So what are the changes from proerythroblast to early normoblast? So there will be decrease in size of the cell. So there will be decrease in the size of the cell. So what is the size here? It is about 15 to 20 micrometers. It is about 15 to 20 micrometers. Here, comparing to proerythroblast, the nucleolus get decreased in size. Yes, here you can see the size of the cell is decreased. It is about 15 micrometers. So the nucleus is also get reduced in size. The nucleoli is reduced, nucleus is reduced, and the chromatin. The chromatin compared to the first stage, here it is very densely compacted. Yes, it is densely compacted. Chromatin is the dense chromatin network is formed. So here also there is no appearance of hemoglobin. Hemoglobin is not appeared yet. Yes, but it is started synthesizing proerythroblastic stage, and it is not appeared in proerythroblastic stage. It is not appeared in early normoblastic stage. And here the cytoplasm stains basophilic. Yes, the cytoplasm stains basophilic. Now let's see the next stage, that is intermediate normoblast. Yes, intermediate normoblast. The cell is further decreased in size. The cell is further decreased in size. The more the condensation is formed, more the condensation of chromatin network. Yes, as compared to the first step, the chromatin is even more condensed. Now there will be appearance of hemoglobin. Yes, this is the main point you need to remember. Yes, there will be appearance of hemoglobin, that is polychromatin. Hemoglobin gives the acidic nature to the cytoplasm. Yes, stains acidophilic. So both priorly basophilic is present. Now hemoglobin is adding, it gives a polychromatic nature. And now the late normoblast. Yes, it is comparatively even more decreased in size, reduced in the size of cell, even the nucleus. The nucleus here is appeared as ink spot nucleus. Yes, the nucleus is appeared as ink spot nucleus. So the nucleus get reduced, it gets disappearance as it is getting matured, it gets disappear. So the nucleus disappear by pycnosis. Pycnosis, it is a process. Yes, the nucleus is disappeared, and increase in hemoglobin concentration. Yes, there will be increase in hemoglobin concentration and reaching reticulocyte. Yes, now our cell reached reticulocyte stage. Now here there will be complete absence of nucleus. Here the nucleus is completely absent. Yes, now there will be a decrease, obviously decrease in size of the cell. It is about 7.5 micrometers. Here it stains basophilic, the cytoplasm stain. The cytoplasm stains what? Basophilic. Here the reticular network is formed, or reticulum is formed in the cytoplasm. Here in this stage, the reticular site, the cells enter into the bloodstream. Here are these cells are the these are the mature, obviously the matured, almost mature cells, they enter into the bloodstream through capillaries at this stage. Yes, and let's see our hero. Yes, matured RBC. These are matured erythrocytes. The size is about 7.2 micrometers. It is about 7.2 micrometers. Here the nucleus is completely absent. Yes, there will be even more increased in Hb concentration, giving red color pigment to the RBC. Yes, it contains hemoglobin, very high hemoglobin, which gives red color pigment to the cell. So there will be complete disappearance of reticulum, which is seen in the reticulocytic stage, and the RBC reaching biconcave shape. Yes, there are some advantages, benefits why the RBC is biconcave. It is due to equal distribution of oxygen and easy removal of substances and easy addition or removal of the different substances and transport of gases. So the biconcavity helps in such a way. And let's see what are the factors that are necessary for erythropoiesis. Yes, there are different factors that helps in the process of erythropoiesis. See, there are some stimulating factors. There are some stimulating factor which which facilitates the erythropoiesis, in which the first one is hypoxia. What is hypoxia? It is the decreased concentration of oxygen availability by blood to the tissues. Yes, there will be decreased oxygen availability to the tissues. So the hypoxia stimulates the process of erythropoiesis. Yes, it stimulates the process of erythropoiesis by inducing secretion of erythropoietin. So the hypoxia, when a certain tissue is inadequate, yes, let's say that is decrease in oxygen, it attained hypoxia. So this stimulates erythropoiesis. Yes, how? It induces secretion of erythropoietin. Erythropoietin is a hormone released by the kidney. Yes, it is released by kidney, which helps in the process of erythropoiesis. Yes, now the next point is, the next factor that is necessary for erythropoiesis is erythropoietin. Obviously, it is released by kidney. As I say, it is secreted by peritubular capillaries of kidney. It is secreted by peritubular capillaries of kidney. So, what are these functions? What are the functions of erythropoietin? Yes, it helps in production of proerythroblast. What is the first stage of erythropoiesis? It is a proerythroblast. Next function is development of proerythroblast. So it has two functions. It has production of proerythroblast and the development of proerythroblast. And after attaining after reaching reticulocytic stage, they are also released into bloodstream. The erythropoietin helps in production and the development and the release of RBC. Yes, and the third factor is thyroxine. It is a hormone. Yes, the thyroxine, it is a hormone which enhances, which increases the rate of process of erythropoiesis, which accelerates the rate of erythropoiesis. And the fourth factor which helps in the erythropoiesis is sex hormones. What is the role of sex hormone? This is not that important as it is important in hypoxia erythropoietin. It is as the testosterone, it is a male hormone which helps very mildly in the erythropoietic action after attaining puberty. Yes, it has mild erythropoietic action after attaining puberty in the males. Yes, so these are the stimulating factors. One is hypoxia, one is erythropoietin, thyroxine, and the role of sex hormones. Yes, now what are the maturation factors? Here the RBC is formed. Now what are the maturation? In order to mature, what are the maturation factor? The first one is vitamin B12. It is also known as cyanocobalamin. It is an extrinsic factor. Yes, the vitamin B12, it is an extrinsic factor. Yes, it is absorbed from diet. It is absorbed from gut into blood through the presence of intrinsic factor of cancer. Yes, the vitamin B12, it is also known as cyanocobalamin. It is an extrinsic factor. It is absorbed from the gut, from the diet in the presence of only intrinsic factor of Castle. So for it helps. What is the functions of vitamin B12? So it helps in the synthesis of DNA in RBC. Yes, it helps in the synthesis of DNA in RBC. So if the vitamin B12 is absent, if we have the deficiency of vitamin B12, it leads to anemia. What anemia? It leads to pernicious anemia. It leads to pernicious anemia. So the next factor is intrinsic factor of Castle. So intrinsic factor of Castle, it is produced by parietal cells of gastric glands. Yes, the gastric glands, the stomach containing gastric glands, these are aligned by the parietic cells which secretes intrinsic factor of Castle. So which is in the first point we have completed B12 absorption, for which we need intrinsic factor. So here the intrinsic factor function is to absorb vitamin B12 from the diet, from the GIT. So the next factor is folic acid. Yes, folic acid. It is important for the maturation of RBC, as it is the function of folic acid, same as vitamin B12, in which it is involved in the maturation of RBC. So the deficiency of folic acid leads to megaloblastic anemia. Yes, it leads to megaloblastic anemia. So next, let's see the factors that is necessary for the synthesis of hemoglobin. Yes, there are different factors. Let's see some factors which helps in the synthesis of hemoglobin. The first one is first class proteins and amino acid. First class proteins and amino acid, which helps in the synthesis of protein part, which helps in the synthesis of protein part of hemoglobin, that is globin. Yes. Now iron is also involved in the synthesis of hemoglobin, where it involves in the synthesis of heme part, heme and globin, right? So the iron is important for the synthesis of heme. This iron is absorbed from the gut, from the diet we take. So the cobalt is necessary for the absorption of iron for the synthesis of heme. So here the cobalt is involved in the absorption of iron for the synthesis of heme. So there are some other factors like cobalt, nickel, and vitamins like vitamin 2, vitamin B2, vitamin C, vitamin B6, etc. So these are the factors that helps in complete erythropoiesis. There are factors for stimulating factors, there are maturation factor, and there are some factors which are necessary for the synthesis of hemoglobin. Yes, there are 6 stages in the hemoglobin: the pro-erythroblastic stage, early normoblastic stage, intermediate normoblast, late normoblast, reticulocyte, and mature RBC. In the pro-erythroblast, you need to remember that it is the largest cell, it is a megaloblast. There is absence of nucleus. These are the main important in the pro-erythroblast. In the early normoblast, it is important that it is basophilic. Yes, it is a basophilic in staining cytoplasm. In the early, intermediate normoblast, you need to mention that the HB starts appearing. Yes, the hemoglobin starts appearing. In the late normoblast, you have to mention that the nucleus disappear. The ink pot nucleus get disappear. In the reticulocyte, you have to mention that there is complete absence of nucleus and the formation of reticulum in the cytoplasm. In the matured RBC, obviously you need to mention absence of nucleus, the size of nucleus, and the increased hemoglobin, obviously, and biconcave and reticulum is disappear. Yes, this is a complete brief discussion on erythropoiesis. It can be asked for a short answer or a long answer. And if it is a long answer, you need to mention the factors that are helping for the erythropoiesis and HB hemoglobin synthesis. And if it is a short answer, you can just write introduction, definition, and explain the stages with the diagram. Yes, with the diagram you can explain for short answer. Yes, this is the complete discussion of erythropoiesis. If you like the video, please do like. And if you have any doubts, please comment in the comment section. Subscribe the channel and support us. Thank you.